I want to tell you, before we start this session, I just want to tell you that often you're given a pedigree and you're told there's just one proband. And what it is, the investigator has just called the first proband, he's just called that a proband. That's really an index case. So you've got to be very careful to find out whether or not you have single ascertainment or whether the family was ascertained on more than one proband. For those of you who are interested, it was Lyndon Eves who was the person who considered the possibility of the analysis, the parameters of interest may depend on family structure. Also, I want to tell you, I referred to a book on by Ginsberg, Malkin, and myself, and where that explains how to allow for ascertainment in pedigrees. Okay, so now let's go to talk about genetic epidemiology. There's several steps in genetic epidemiology. So there are 4 steps. There's, first of all, you can look for aggregation, then you can look for segregation, and then you can look for co-segregation. Those are the 4 steps. Now, for a classical rare monogenic disease, familial aggregation first, then segregation, then you put linkage analysis, which is co-segregation with a marker whose location on the chromosomes is known. And then you go to what is known as positional cloning. Well, now, the term positional cloning was first used in a paper or presentation by Collins, which I can't find in his CV, but I remember reading this and thinking, oh, all he's talking about is linkage analysis. If anybody knows where that paper is, please let me know. But I know I read that long, and it's not in his CV. And people talk about Francis Collins introducing the term, and I don't know where. It must have been at a meeting or something. So let us suppose we are studying a complex disease. The first thing is to determine whether you have familial aggregation and possibly a genome scan. And then you go to either linkage analysis or association analysis. And you search for a candidate gene or region, which is a region for a candidate loci or linkage disequilibrium. Let's consider aggregation. Does the disease aggregate in families? Do parents, offspring, and siblings of affected persons have a higher risk of disease? Do more distant relatives of affected persons have an intermediate risk of disease? Do spouses of affected persons not have a higher risk of disease? These are all questions you want to know. Let us consider segregation. Now, you may have affected and unaffected, or you may have 2 distributions. And those 2 distributions may be determined by dominant or recessive allele. You also may have the possibility of 3 distributions. So the question is, can the dichotomy or trichotomy be explained by Mendelian segregation? Let me talk to Yunju. Yunju, can you, if people ask a question, will I be able to see them or hear them?
Yes, you will be able to hear as long as they unmute, and then
okay.
See, I will explain it during the break.
Okay. So let us consider various segregation analysis models. First of all, there's the transmission probability model, and then there's the mixed model, major gene and polygenic. And these were unified, making the mixed model and transmission probability model. They were unified by Laluelle back around, I don't know, whatever that, I can't see that date, or about 1983. And you have to understand that until, let's see, throughout the 70s and 80s, there was a lot of discussion about whether one should use the transmission probability model or the mixed model. And I was favoring the transmission probability model. Newton and his colleagues were offering the mixed model, major gene plus polygenic. And Laluelle put the two together. He was a postdoc working with Newton Morton. And then about 1983, the regressive models came up. Now, the transmission probability model was first introduced in the Elston-Stewart paper. The probability that an individual has genotype UI, given the two parental genotypes, U sub Fi and U sub Mi, that probability is a transition probability where U is the genotype. On the other hand, the probability that the AA individual or AB individual or BB individual, the probability that each of those transmits a capital allele, these are transmission probabilities. These are the ones that we look at in the transmission, in the transition probability or transmission probability model. These are the parameters of interest. Now, for two alleles at one autosomal locus, there are 12 functionally independent transition probabilities. There are 6 mating types, each with 3 probabilities that sum to 1. So that leaves you with was it 12? There are 12. So there are 12 functionally independent. 6 3s are 18, but they add to 1 in 6 cases. So you have 12 functionally independent transition probabilities. Now we need to talk about likelihood. So the likelihood of a set of parameters given a set of data is proportional to the probability of the data given the parameters. And the probability of the data on all the members of a pedigree can be written as a function of transmission probabilities. So that's what we do. We look at these 3 functions, the transmission probabilities, TOR AA, TOR AB, and TOR BB. And if there is Mendelian segregation, these have the probabilities 1, half, and 0. So that's what we're looking for. In the case of co-segregation, we have to remember that chromosome segments are transmitted. Co-segregation is caused by linked loci. And remember, and this was Andreas Ziegler who made this wonderful expression, loci are linked, alleles are associated. You will never forget if you remember that. Loci are linked, not alleles. Alleles are associated. So in co-segregation, we determine whether markers are linked to or inherited along with the predisposition to the development of disease. And if so, such markers help define candidate regions wherein there may be causal genes. Now, I want you to please, if you don't understand anything, you should speak up, because if you don't understand what's coming in the next slides, you will not understand a lot of the S.A.G.E programs. Don't be afraid to speak out. Or if you put in a chat note, Yunju will let me know. Contrast the situation where there's no linkage In other words, the recombination fraction theta is a half. That's what we're going to look at first. Now, if you look at this picture on the left, you notice that, by the way, the two dots in the middle are the centromere. You'll notice that the A locus and the B locus are far away. So theta is equal to 1/2. So the probability you get an capital A, capital B haplotype being transmitted is probably 1/2 times the probability there's no recombination, which in this case is 1/2. So that probability is 1/4. The probability of having a gamete transmitted capital A with lowercase b, that's 1/2 times, but now you have to have a recombination. But if the recombination is 1/2, that's 1/2 times 1/2, it's 1/4. Similarly, for the other haplotype where there's been recombination, little a and capital B, it's 1/2 times 1/2, 1/4. And for the little AB, the little AB, the right bit, for that to be transmitted, the probability is going to be 1/2 times the probability there's no recombination, and that again is 1/4. But contrast this now with linkage. And let us suppose, for example, that the recombination fraction is 0.1. Now look at these, the probabilities of these haplotypes, which is 1/2 1 minus theta, 1/2 theta, 1/2 theta, and 1/2 1 minus theta. And you see immediately when theta equals 0.1, let us say, these probabilities are no longer quarter, but 0.45, 0.05, 0.05, 0.45. So that's the whole point of linkage analysis. You find that the haplotypes do not segregate independently to the offspring because there may be recombination. And remember, a recombination is an odd number of crossovers. Now, what is genetic distance? Well, now, genetic distance is basically you make up a genetic map, and a genetic map distance between two loci is defined as the expected number of crossover events between the two loci. It's not the expected number of recombinations, it's the expected number of crossover events. That's the genetic map distance. So 1 Morgan is the distance in which we expect exactly 1 crossover event. And 1 Morgan, or 100 centimorgans, makes 1 Morgan. Morgan was a Drosophila geneticist around, I don't know, about 1920. And he was the one who basically worked out, for example, that just by analyzing phenotypes, no molecular stuff at all, he worked out that there are 4 chromosomes in Drosophila. He found there were 4 linkage groups. So what is a map? A linearly arranged set of loci with map distances between them is called a genetic map. Remember, a genetic map is in Morgans, crossover events. What we see, though, are recombinations. The autosomal human genome is about 38 Morgans long, sex average, 46 for females and 29 for males. That's the latest information I can find. It seems to vary from year to year. But the whole point is, if you have about 180 markers spanning the genome at 20 centimeters apart intervals, then you can span the whole genome by linkage analysis. Now, a rule of thumb is that 1 centimorgan is on average about 1 million base pairs. Okay, that's only an average. All right, let's consider genetic markers. A genetic marker is a measurable trait that is genetically determined, can be accurately classified, and has a simple, unequivocal pattern of inheritance, and is polymorphic. That's the basic definition of a genetic marker. So what types of genetic markers do we have? Well, in the 1940s, we had the blood groups, like the ABO blood group, the rhesus blood group, many more. Then around in the 1960s, we started finding enzymes. Harry Harris at the Galton lab, he found you could look at these enzymes and run them in gels. In that very early on, it was starch gels, then it became other better gels, and you could see bands, and they were used as genetic markers. In the 1980s, we start having restriction fragment length polymorphism, RFLPs. Then in the 1990s, we started having microsatellites, which are repeats of CA repeats in the DNA. And then finally, around the 2000s, we started having single nucleotide polymorphisms, or SNPs. So what are the goals of linkage analysis? Well, evidence for linkage between a known marker, which you know is genetic, and a putative genetic locus for a disorder is generally taken to be the ultimate statistical evidence for a genetic component in disease etiology, i.e., evidence supporting the existence of this locus, and for there being somewhere in that neighborhood a locus that is monogenic usually. The whole point is that it's difficult to conceive of environmental factors that could mimic genetic linkage between two traits, especially when the genetic marker is known to be caused by segregation at a single locus. Let me give you an example. Just suppose, and this is not true, suppose you've got a disease, there's a particular disease, and let us suppose we look at the ABO blood group as a marker. Now, if there is no linkage in any pedigree, the disease would randomly follow the A allele or the B allele or the O allele. However, if there is linkage, you'll find in some pedigrees, the disease follows the A allele. In others, it follows the B, and in others, it follows the O allele. So if you have intrafamilial association, but no association across the whole population, that is typical of linkage. Now, you can work out a very complicated gene-environment interaction for this, but it's so unreasonable as to be very unlikely. On the other hand, a genetic explanation is the simplest explanation there is. If you find intrafamilial association and no association in the whole population. So localizing of a gene locus, this provides a means of finding a locus solely on the basis of the chromosomal location without regard to its biochemical function. And this is the basis of the positional genetic approach. Once you've found linkage between several pairs of loci, once you've detected that, a genetic map can be constructed specifying the locations of these loci inferred from recombination fractions between pairs of loci. And that's what Thomas Hunt Morgan did for the Drosophila years and years ago. Localization of a gene locus is followed by attempts to clone the gene and characterize or sequence it and determine its function and or the nature of the defect it causes. But remember, we don't clone genes, we clone alleles. So those people who you talk about the ABO gene, there's no way you can clone the ABO gene. All you can clone are the particular alleles. So let's consider linkage between a marker and a disease. There's intrafamilial association, typically no population association, but there is population association if the two loci are very, very close, the marker locus and the disease locus. I hear no questions. I want to warn you, if there's no questions, we will finish so early. I'm used to having lots of questions. Okay. Now, there are two main types of linkage analysis. But all methods of linkage analysis require the assumption of a probability model. You have to have. If you can do any statistical analysis whatsoever, you have to have a probability model. So with respect to the mode of inheritance, the trait being linked to one or more markers, however, the analysis may be either model-based, you specify the disease genetic model and only estimate the recombination fraction, or model-free, you do not specify the disease genetic model and estimate functions of the recombination fraction and the genetic model parameters. In these model-free methods, we rely on identity by descent between sets of relatives. Now, be very clear about this. You have to have a probability model. If there's a probability model, there have to be parameters, okay? So I say model-free when there's no parameters other than maybe something in the model that has some function of the recombination fraction and other genetic model parameters. Now, in the literature, you will find these two kinds of model called parametric and non-parametric. Now, if any of you are statisticians, you may know it doesn't make much sense to talk about parametric and non-parametric. And some people have said it's impossible to have non-parametric analysis. There must be some parameters, or what are you doing? What are you estimating? What are you testing? The whole point is, it is better, and a lot of people I know, we say in statistics, we don't use non-parametric statistics, we use distribution-free methods of analysis. And that's why I make the distinction model-based and model-free. It doesn't make any sense to say non-parametric, because even in a model-free analysis, there is some parameter. You may not know exactly what it is, but there's some parameter that is being estimated, and in fact, you're testing whether it has some null hypothesis value or not. So what are the model-free linkage methods? Model-free linkage methods do not require specification of a genetic model for the trait of interest. They do not require a precise knowledge of the mode of inheritance controlling the disease trait. That's the whole point. Yes, there are parameters somewhere, because if you're estimating or testing, you must be estimating or testing a parameter. But it's model-free in that there is no genetic model that is being assumed. Model-free linkage methods are often computationally simple and rapid. And for that reason, model-free linkage methods can be used as a first screen of multiple markers to identify promising linkage relationships that you can then subsequently confirm by other, maybe model-based methods with more power. So what are these model-based linkage methods? Well, if you're going to use a model-based linkage method, you must specify or at least estimate certain parameters are, for example, the founder disease and marker genotypic frequencies. Either you've got to specify them or estimate them. There's also the penetrance function, the probability of the disease given genotype. Either you specify it or you estimate it. For example, consider a disease that with respect to a locus that with respect to disease is dominant with incomplete penetrance and no sporadics. And the probability of disease given, say, capital A is 0.8, given capital A little a is the same because it's dominant, and the probability of disease given little a is 0. The fact that the first two probabilities are not 1 means you have incomplete penetrance. The fact that the disease given you have little a is 0 means you have no sporadics. So that's just an example. Transmission probabilities, in the case of linkage analysis, they are functions of the recombination fraction, theta. So what we are going to do in a model-based linkage analysis, we're going to use likelihood to estimate the parameters of interest to us that we're not willing to specify. And so that likelihood, and in particular, we're most interested in the recombination fraction. So here we're talking about the likelihood given some data, phenotypic data, X. And the likelihood is proportional to this equation that you can see here. First of all, forget the summation sign. We have the probability of the data given the genotype, which is a penetrance function, multiplied by the probability of that genotype. But we don't always know the genotypes. For example, we may have phenosets. So you have to sum over all possible pedigree genotypes in order to get the likelihood of the recombination fraction. Likelihood ratio. This is very important. The likelihood of theta given a set of data is proportional to the probability of the data given the recombination fraction theta. In fact, this is true for, I mean, it doesn't have to be recombination fractions. This is any parameter. The likelihood of a parameter or set of parameters given the data is proportional to the data given that parameter. But now, if you look at the likelihood of the data divided by the likelihood under the null hypothesis, a particular value, in the case of the recombination fraction 0.5 given the data, then you can equate this probability. The ratio of the two likelihoods is equal to the ratio of the two probabilities. That is how we can get around things. But the ratio of the true probability is not the same as the probability of the recombination fraction given the data divided by the probability of the recombination fraction is a half given the data. What it is equal to is that same like a ratio, but you've got to pre-multiply it by the what are called the prior probabilities, the prior probability of the recombination fraction divided by the prior probability, it's 0.5. So long as you look at likelihood ratios, you're in good shape. Likelihoods by themselves don't mean much, typically. So what is the standard or Lodge score method of model-based linkage analysis? The null hypothesis is that recombination is 1/2, no linkage, versus the alternative that the recombination fraction is less than 1/2. Now, this is very simple But you know, the recombination fraction could be sex specific. There could be two parameters there. It could be that both are a half versus both are less than half. I'm just taking a simple situation here. The log, which stands for log of the odds, that log score is Z, which is the summation of all the logs for the individual families, with summation where I is the we're talking about the ith family here. We sum over all the ith families on the assumption that these families are independent. And so we've, instead of Zi, we put the log to base 10 of the likelihood for a particular recombination fraction given the data, divided by the recombination, the likelihood for the recombination fraction equals a half given that data. So Zi is the log score for the ith pedigree. Okay, and the likelihood for a theta star given the data is the likelihood of the recombination fraction when you stick in theta star for the ith pedigree. Now, you'll notice that this thing called log of the odds is a likelihood ratio. It's put to base 10 so people can understand it better. That was the idea. But you have to understand, now why was it called a log of the odds when it's a likelihood ratio? Because those two things are different. Why? Well, those of you who are interested can go and find a paper, by, forgotten the name, begins with B. But the point is, he talked about forward logs and backward logs. And what the log he's talking about here is a backward log. It's a likelihood ratio. So that's the history of why it's called a log when it's not at all the log of the odds. It's the log of the likelihood ratio. Don't be fooled if you know what odds are. This is not the log of the odds, but the log of a likelihood ratio. And to base 10, which people could understand because they know that, you know, log to base 10 of 100 is 2, of 1,000 is 3. So you can understand that magnitude much better than if it were to base E, the natural logs. So let's consider a single marker. We call this in S.A.G.E. a single marker linkage analysis. But what the classical people called it was a two-point linkage analysis because there were two loci involved. We call it a single marker because there's only one marker. The other trait is going to be it's going to be a trait like disease or not treat or another locus or what have you. So what used to be called, or most people call, two-point linkage analysis, we call single marker analysis. Typically what we do is we calculate the likelihood ratio of theta at different values, 0, or we calculate actually the log score at theta equals 0, 0.05, 0.1, 0.2, 0.3, 0.45. But, and this came later when people realized, oh, we don't have to look at those values only. We can actually estimate theta. We can find the maximum likelihood estimate of it. Theta, as I said before, may depend on the sex of the parents. It's usually larger for females than for males, but the opposite tends to hold in telomeric regions. Telomeric regions are the ones at the ends of the chromosome. So remember that the log to base 10, the log score are logs or log scores, but there are logarithms of likelihood ratios and not logarithms of odds for linkage. And the real point about them is if the families are independent, you would multiply the likelihoods, so you can just add the logs because they are on a logarithmic scale. The maximum log score, which in other words, you evaluate it at the maximum likelihood estimate of the recombination fraction, measures the amount of evidence for linkage. So z theta hat is log to base 10 of theta hat divided by the log when theta equals 0.5. And similarly, if you had two thetas, you'd put at the bottom 0.5 for the one sex and 0.5 for the other sex. That measures the amount of evidence for linkage. Now, traditionally, and I'm talking about going back to the 1950s, 60s, and 70s, if the maximum log score was greater than 3, that was proof of linkage. And if it was less than -2 in any region, less than, it was proof of there not being linked within a recombination fraction of whatever that theta is. The problem now is, in those days, we didn't worry about the fact that you could have thousands, hundreds of thousands, or even millions of markers. So this was all on the assumption that you were doing linkage analysis with one marker at a time. Now, multi-point analysis finds the position along a chromosome that maximizes the likelihood using all the markers, as opposed to single point, it's multi-point, okay? We use the word single point, which it says there's only one marker, which years and years ago, people called 2 points because there were 2 loci involved, one for the disease and one marker. We use single point there as opposed to multi-point, where we're looking at a span along the chromosome of many markers. So when we have many markers together, each marker gives information at every point. But when we perform multi-point analysis, it assumes no interference and linkage equilibrium. And the recombination fraction between 2 consecutive markers is related to genetic distance. And remember, genetic distance is not physical distance is not genetic distance. They're not the same. Make that distinction. Physical distance is in base pairs. Genetic distance is in Morgans, or the crossing over. Well, actually, not the crossing over. It's in the it's in the crossing over, but we can only estimate the recombination fraction. You can look at this paper where you'll, I think a lot of things are explained. So, what are the founder genotypic frequencies? Consider two loci, let's say a trait with alleles capital T and lowercase t, and a marker with alleles M1 and M2, just for simplicity. There are 4 haplotypes: TM1, TM2, the capitals, and then the lowercase tm1, the lowercase tm2. And so you have 5 by 4 phase diplotypes. Let us look first, before we talk about diplotypes, and what are often, to stress it, are called phase diplotypes. Let us look at the 9 two-locus genotypes. Remember, at one locus, with one allele, you have two alleles at one locus, you have 3 genotypes. So we have 3 genotypes at the T locus and 3 genotypes of the M locus. So here is the 3 by 3 table of the 9 two-locus genotypes. These are the two-locus genotypes. Notice the one in the middle, the yellow. That corresponds to two what are called diplotypes, because there it matters what the phase is, and the phases means which allele comes from this, which parent, mother or father. So let's consider that middle two locus genotype, capital T, little t, M1, M2. This double heterozygote corresponds to two different diplotypes, the TM1 over lowercase tm2 and the little tm1 over the capital TM2. What this indicates is, on the left, capital T and M1 came from one parent, and little t and M2 came from the other parent. On the other hand, on the right, the little t and the M1 allele came together from one parent, and the capital T and the M2 came from the other parent. So if we know which parent the alleles came from, the sets of the parents, we have two different diplotypes. They often call phase. These are called the two different phases of linkage. And we usually assume these are equiprobable. They don't have to be, but this is a usual assumption. We also assume Hardy-Weinberg equilibrium at each locus, whether it's true or not. We make these assumptions, and these are difficulties if they are not true. We also assume that the frequency of the haplotype XY is the product of the allele frequencies X and Y, where X and Y are not allelic. They're on different loci. And so we are assuming what is known as linkage, allelic, or gametic equilibrium. These are all assumptions, some of which people have tried to tackle by different ways, but the usual typical method of linkage analysis, model-based, is what I've just described. However, we have to look at penetrance functions. We usually assume that the probability of the trait phenotype and marker phenotype given the two locus genotype is the same as the trait phenotype times the probability of the trait phenotype given the trait genotype times the probability of the marker phenotype given the marker genotype. Remember, I'll stress this again, we never know the marker genotype. All we know is the marker phenotype. So we usually assume that. The trait locus follows a model either estimated by segregation analysis or guessed. It used to be years ago guessed. All we will do a segregation analysis and then somehow assume some model from the result of a segregation analysis. The marker locus often displays a one-to-one relationship, codominance between genotype and phenotype, but it doesn't have to. For example, at the ABO blood group, we already know there are phenosets, which suggest that whatever sometimes when you see a phenotype, it could be due to more than one genotype. Phase. Phase is the very important idea. So the phase of the transmitting parent can only be determined if you have knowledge of the parent's parents. So you have to have more than two generational data to know phase. If you just have a SIBSHIP, you can't know where the grandparental alleles came from. You've got to have more than two generations to do that. So what does this mean? When there's no information about a phase, I don't care what people might say, the likelihood, log likelihood, and log, the log likelihood ratio, are all symmetric about theta equals 0.5, recombination fraction. Now you may say, but I know it's not between 0.5 and 1, so it can't be there. But that's your assumption. The data cannot tell you that. All the data says it's either the left curve or the right curve, either the continuous, the one that's the white line, or the dashed yellow line. You don't know. It's your assumption that the recombination fraction is less than 0.5. The data cannot tell you that. So SIP-SHIP data and nuclear family data contain no information about phase. You have to have pedigrees. SIP-SHIPs and family, nuclear families, just the two parents, can give you no information about phase. But cousins, you have to know, are 3-generational data with the first 2 generations missing. So they're essentially 3-generational. So cousins can give you information about phase. SIP-SHIPs, nuclear families cannot. So that's a brief description of a linkage analysis. Now let us consider association analysis. Well, before I go on, are there any questions about linkage analysis? Because at the rate I'm going, we're going to finish very early, unless people have some questions. No questions? You're all shy. All right. So either you know it all or you don't understand a word of it. Okay. I suspect most of you know much more about association than linkage, because that's the modern thing. So there are several causes of association between a marker and a disease. You can have just chance, okay? And a lot of what we do these days in association analysis with hundreds and hundreds of thousands of markers, we have to worry about chance. And that's why we usually require a very, very low p-value. There could be stratification, population heterogeneity, and I'll talk about that in a minute. There could be selection, genetic selection, you don't know. And there could be tight linkage. Of course, you hope you're going to get tight linkage. But before you even consider that what you found is tight linkage, you have to consider the other possibilities. And lastly, you may find pleiotropy. That means that someone asked me once, and I gave this course, okay, you say you found tight linkage. How do you know you haven't got pleiotropy? Well, think about it. What does pleiotropy mean? It means that marker locus itself actually causes the disease. So what do you care? You don't care if it's pleiotropy. That's even better, right? You found the disease locus. Why would you worry that you may have found pleiotropy instead of tight linkage? So you have to think what you're doing here. Heterogeneity and stratification. I want you to first look at this table with 2 subpopulations. Okay, we have two 2-by-2 tables. All right. So in subpopulation 1, you'll see the numbers you have. You have 1,000 people in each population. In subpopulation 1, you'll see the entries are 50, 50, 450, 450, total of 500 for the antigen present, 500 for the antigen or the A allele, whatever you want, whatever it is. Say the A allele present, the A allele are missing, and then you've got disease present or disease missing. Now, what I want you to notice, the 2 tables are different. They both have 1,000 individuals. The important thing to note is, what is the probability of disease, D+, given the allele A or antigen or whatever? Okay, the allele A+. Look at subpopulation 1. Look at the first column. The probability of disease, given you have A+, is, if you have A+, you know that 50 out of 500 people have the disease. Okay? and given they have A minus, you see also 50 out of 500. In other words, and you also notice that there is no association in this table because it's the same. By the same token, if you look in subpopulation 2, under the A plus column, you see disease occurs once in 100 cases, total of 100. And in the case of the A being absent, you have 9 out of 900. In both cases, it's 1 out of 100. There is absolutely no association in either of these two tables. Another way of looking, you work it out for yourselves later, is to say that 50 times 450 minus 50 times 450 in the first table is exactly as 1 times 891 minus 9 times 99 in the second table. Now let us put these two subpopulations together. Now we have the total population. All right, you can do the math. First, top left square, 50 plus 1 is 51. 50 plus 9 is 59. 450 plus 99 is 549, and so on. We have 2,000 individuals in all. Now let us look at the probability of the disease there, given that the A is there. In the left-hand table, it's 51 divided by 600, which is 8.5%. In the right-hand table, on the other hand, it is completely different. It's 59 over 1,400, and that's about twice as big. So clearly there is association. This is association due to homogeneity stratification or stratification. You have to worry about it because when you're doing association studies, this is what you may be finding. So now let's talk about linkage disequilibrium. We talked about it before. Let's look at it another way. Here are some ancestral haplotypes. Okay, I've just got 4. Typically, you have hundreds or thousands of them. They're happily in the population, and they're going from generation to generation, and they're, they're just happily being transmitted. Now, because of crossing over, after a bit of several generations, maybe 5, 6, 8 generations, whatever, what you get is you get them all mixed up because of the crossing over. Now, some of these individuals are affected with the disease, some are normal. Now, just look at this region here. You'll notice the affected all have the same little bit of ancestral chromosome, whereas the normals have a random sample of it. So that is due to linkage disequilibrium. And that is the basis of association analysis when used for genetics. If you find the association, if the association is due to linkage disequilibrium, that means that little region on the chromosomes is, in fact, very close to, or possibly it is, the locus at which affected and normal is segregating. Stratification, heterogeneity. What are we going to do about it? Well, there are 3 solutions. One is to use a homogeneous population. Well, suppose you don't have a homogeneous population. Then you can use family-based controls, because if they're family-based, they're much more likely to have the same kind of, they're much likely to be more homogeneous. The other thing you can do is use genomic control. And, we'll talk about these 3 now. So in association study samples, let's consider 2 extremes. you may have unrelated cases and controls, which will detect causal loci, also detects linkage disequilibrium, also detects heterogeneity stratification. So if you have heterogeneity stratification, you can use what is known as genomic control. You estimate, or you can estimate ethnicity with other markers. And genomic control, I'm not going to go into it, but it's important to read it up. There's a parameter for it, which you look at and you do all these nice plots to see if it's all okay. Estimating ethnicity with other markers, that's often called doing principal components analysis, PCA analysis of the markers. Or you can use cases and controls match for ethnicity. You can have what are sometimes called pseudo-SIBs. They're not really SIBs, they're close friends. You think they were brought up together, they're very similar in everything except their genetics. Or you can use other relatives. Important that, and by the way, if you read the paper down the bottom here, Devlin, Roden, and Wasserman, they will explain genomic control in it. The whole point is that causation implies association, but the converse is not true. It doesn't go the other way. Just as correlation does not imply causation. Causation will give you correlation, but the presence of causation doesn't imply correlation. Same thing for association. If you have causation, you will get association. But if you have association, that does not imply causation. So what's the challenge of complex diseases? Oh my, complex diseases. So what's a complex trait even? I had great difficulty finding out what that meant a long time ago. And I was asked to give a talk about it. I said, I have no idea what a complex trait is, so I'll talk about I think I called it a multivariate trait. Anyway, the whole point about what people mean by a complex trait, it refers to any trait that does not exhibit classic Mendelian inheritance attributable to a single gene locus. That's what people mean by it in genetics. Complexities arise when a simple correspondence between the genotype, genetic endowment, and phenotype, the observable manifestation, breaks down. And so we have to allow for these things. What are some examples of these complicating factors? You may have incomplete penetrance and not allow for it in the analysis. You may have variable age of onset and not allow for it in the analysis. You may have difficulties of phenotypic definition. By the way, the first two, incomplete penetrance and variable age of onset, this we can allow for in S.A.G.E, as long as we have the right sample to estimate what to do about these complicating factors. If you have difficulties in phenotypic definition, there's not much I can do for you. It's up to you to define exactly what you mean. You may have phenocopies. You remember that? That was the thalidomide syndrome. You may have genetic heterogeneity, and you've got to allow for that somehow. There may be other genetic mechanisms such as imprinting. And imprinting, what imprinting means is that the expression of the depends on the expression of a genotype depends on the sexes, the alleles, which allele comes from which parent, male or female. That's imprinting. So what is the best design and the best analysis? Well, I was asked this question sometime around 1990, where there's a lot of questions, what should we do, linkage analysis or association analysis? Well, if heterogeneity and stratification is a non-issue, and we talk about common disease alleles, then there's no doubt about it, unrelated cases and controls for association analysis is the right way, and perhaps a genome scan. which is what people do these days. They have hundreds and thousands of markers, look at the whole genome. There's no doubt about it. That's the best if in the simple case. But if you have heterogeneity and stratification, and they could be a real issue, and you have rare disease alleles, and you really want to look at the whole genome, then large extended pedigrees, type all the individuals, founders and non-founders, with equispaced markers for linkage analysis. That is the best design. And I would suggest that don't forget, there's the cost, the burden of multiple testing. The more you test, the bigger the sample size you need. I suggest a wise investigator, like a wise investor, will hedge their bets with having a judicious mix of these two. So I strongly suggest that you don't automatically go for one or the other if you don't know. Back around 1990, I was asked, which should you do? And what happened, this was by the S.A.G.E Institute. And what they decided to do was association analysis. And they learned a lot. But as you know, there's a lot of trouble with association analysis, and there is the burden of multiple testing. And there's been, first of all, association analysis has led to a lot of good results. Unfortunately, it's also led to a lot of bad results. Okay? And that you have to remember. Now it's already time for break. I suspect we've gone far too fast because nobody's asked any questions. It is now 3:30. Does anybody have any questions at all? Are you brave enough to ask questions? I don't hear anybody. Should I be hearing somebody?
If anyone has a question, you can ask. You can unmute or turn on your video. Otherwise, I guess we can have a short break and then resume at 3:45. So we gives about like a 15 to 20 minutes.
All right, we start again at 3:45.
Yes.
What was the time that we had reckoned it would be, just so I know?
Actually, 3:45 to 4 o'clock is the break, but we are about like a 15 minutes.
All right, so I tell you what, we'll come back at 3:45, and maybe at that time you have a lot of questions. I don't know whether you've all disappeared. I can't see you on my screen, whether there's more of you here. But if you have questions, please bring them then. Otherwise, we'll just have a break. Okay?
Okay.
There's a question in the chat.
Oh, is there anything in chat?
Oh, there it is. Sorry, I just noticed it.
All right. What do we have in chat?
Okay. For complex disease, do you suggest GWAS? For rare disease in families, do you suggest linkage studies?
Well, as I said, it would so there's two possibilities: rare, common, complex, monogenic. That's a fourfold table, right? So let me see. There's been pictures published in, I think, in Science about it. If you have a rare disease and it's simple monogenic, then there's no doubt about it, you will do linkage analysis. If you have a common disease and there's heterogeneity to things like common disease, I'm trying to think in my head. Linkages for rare diseases that are simple. Oh, wait a minute, you said complex. Oh my. I thought
complex disease, do you suggest GWAS? For rare disease in families, do you suggest linkage studies?
Yes, I do. If that was the question asked, yes, I think. But for complex diseases, you may, well, it depends. You may get better luck if you do linkage, because you may find several linkage regions. So I'm not sure about it. Certainly for a rare disease, it's more likely to be monogenic. For a common disease, it's more likely to be complex. As I've just said, a wise investigator, like a wise investor, will hedge his bets with a judicious mix. I would think of both. My own preference is always large families. There's so much more information. In fact, we're now realizing the people who are doing association analysis in large, they're doing it and they're getting association signals. A lot of them are deciding, well, now, is it really genetic? We better do something like linkage analysis to be sure, get some families. And that's, that is the best statistical evidence. Now, if you have biochemical evidence good biochemical evidence for the results of association analysis, then you don't need linkage analysis. But if you don't, I would suggest some large pedigrees, because that's where you will find out whether it's truly genetic or not by statistical analysis. I hope that makes sense. Any other questions?
There is another question in here. Is the genetic distance for a given set of alleles constant over generations in a given population?
Say that again, I didn't understand.
So is the genetic distance for a given set of alleles constant over generations in a given population?
I think people are talking about loci, not alleles, right? Genetic distance refers to loci, not to alleles.
Mm-hmm.
All right. So, yeah. Well, I'm sure you can probably find some exceptions. There may be some populations, rare populations, where the genetic distance is different. Genetic distance is certainly racially, is to a certain extent racially determined. Yeah. To what extent, I don't know. I don't know that but typically genetic distance is not dependent. Distances between the loci are pretty constant, but we do have more crossing over in and therefore more genetic distance in populations that are from mixed racial backgrounds. Okay, now if you were to take pure European whites with some pure blacks from Africa, we know it's an interesting fact, we know very little other than pure white populations. We know very little. We have much less information about pure black populations. We have a lot of information about the ethnic crosses, the African American black. We know much more about that. But the pure blacks in Africa, we have very little information about. So I'd be very leery about assuming too much. I don't like too many assumptions, but I personally would expect the genetic map to be similar, quite similar. Okay. I mean, maybe somebody knows better than me and has evidence that that is not true. I don't know for a fact because we don't know so much, but I would expect the genetic maps to be similar, because remember, the maps is between loci, not alleles. Whereas we know that in the different ethnic populations, we have completely different alleles. All right. Any other question? No?